Well, are we there yet? Let's see. So, I will be throwing at you some questions that you may not expect. But I wanted to ask you, how many of you own a house? Well, the house that was in generation in your family, the house that your great-grandfather built. Is it still standing? Is this looking like that? Or it's like this, shiny and new, with the new residents in it? And if that is the case, let me ask you again, is that the same house? Well, you will be surprised to know that this is the biggest promise of tissue engineering. You will ask me, why is it related to the house? Well, all of you are living in your bodies, and in a sense, it's your own house. And let's see if you one day are in need of a tissue of an organ that somehow got damaged or sick, and you need to replace it and repair it. So what the tissue engineering tells you is like, I do the magic, and I will recreate that organ for you to the degree that it's going to be a perfect functional organ that will be safely transplanted into you, and you will enjoy a long and healthy life afterwards. Well, that's the promise. Let's see how close we are to its realization. First, let's talk about it. What is tissue engineering and what is the engineered tissue or an organ? All of you sitting here probably know that we all made out of cells. So if we wanted to recreate something live, we need live cells. Okay, we need live cells. The sources are different, but they all have to be pretty much young cells in their soul, which means that they should have a potential to become any cell that you need at the moment. Those cells called pluripotent cells. And the pluripotent cells are coming in three flavors. It's either embryonic cells, but they come from the embryo. They're either adult cells, and they will, in this case, will be called mesenchymal stem cells, or they're induced cells. Those are even gotten the Nobel Prize, and these are the cells that have been reminded of being young and pluripotential and being capable of doing many things. So we take the adult cells, reminding them that they used to be kids, that they can achieve many things, and then kindly asking them to become another cell of a type that you need. All right. So we've got the cells, let's say we isolated them, we know how to grow them, we put them, amplify them, and all is well. What do we need next? We need a house for them to live in. So in the house that we are populating our cells into, the science is called scaffolds. So it's basically a structure that reminds them of their natural house, and it is made either artificially or it is taken from another animal or a cadaver, or it is bioprinted. So we'll talk about all these possibilities in a minute. Okay, now we've got the cells, we've got the house they live in, and as you all know, if a house is happy house, it must be healthy communications in it amongst its living organisms. So for that, we need to provide them with the bioactive molecules such as growth factors, hormones, enzymes, signaling molecule, and all that happy mix. So that's what the tissue engineering is telling you. Give me these three components and I'll give you a functional organ. Okay, move on to the type of houses that we are producing for those cells. It's a huge field of study right now called the material science, and basically all scientists are now looking for better and better structures and ways to build those scaffolds for cells to have microscopic niches where they can attach, they can still function, they can talk to another neighbors, they can make your whole organ work. And they're made out of the hydrogels, nanofibers, the gas form, salt leached, and whole repertoire, and the field is growing by the hour. Then comes the big news in everywhere, including the tissue engineering, is 3D printing. Well, it was a big boom when the 3D printing came about, because everybody decided that, oh, that's it, we've solved all the problems. We're going to print any organ of any shape we want, and that will be fine. But all is not as easy as we say, because to print a scaffold for a living cell, you cannot use a simple plastic that doesn't breathe, that doesn't transfer those signaling molecules and bioactive molecules, that does not allow for them to grow, to make connections. So if we're talking about the bioprinting, those bioprinters need bioinks, which means that we are making, again, new materials that are supporting the life of live cells. Okay? And that's also is area where you as an aspiring scientist can say your word. So, but then comes my personal favorite. This is called decellularization. Let's think about the word. You're taking something and freeing it from the cells. Basically, you're taking a tissue that is full of the cells and washing away all of the biological content from it, leaving only biochemical carcasses. So the examples of that you can see on the rat, on the heart, and the liver. So it's looking actually very cool. If we look at the process, what it is, it's actually simple in its principle. You take a fully functioning organ and you start washing either through it, perfusing, or putting the organ into a solution of detergent, which is in the everyday language is simply said soap. So if you put your tissue in a soapy solution and keep it there for a long enough time, all the cells get washed out because the cells are surrounded with the lipid membranes, and as all lipids tend to be washed with the soap, so are our cells. And so eventually, depending on an organ, the time, you will obtain that image on the very right for you, which is basically a ghost of an organ. Okay? So, and then let's say we made it all. We made our organ, we found the cells, we put it into that, and we're ready to transplant. So what happens with the transplantation? What happens always with any old age transplantation of the organ? The immune system of the recipient, with all of its cells, the B cells, the T cells, the natural killers, and the macrophages, starts attacking that organ and start eating it up. And most importantly, and that their main aim, when what they're attacking, is the cells. So the idea is that if we wash the cell out of our transplanted tissue, all these cells already will have nothing to do with it, and they will simply disappear. But all of a sudden, we realize that the dendritic cells of the recipient still lingers around. And a dendritic cell is a kind of a cell that goes, talks to everybody, eats up something, and introduces that to the immune system of the recipient. So basically, this is what the dendritic cell is doing, and it is more realistic representation of the dendritic cell. What that cell has is a special receptors that are capable of processing not only the cells that we did not have already in our tissue, but some remnants of something. It could be nuclear or mitochondrial pieces of DNA. It could be fragments of the same very proteins that remains in the scaffold, or they can be some peculiar sugars that if you're using a tissues not from the primate origin. So all of that is making these cells to just stimulate inflammation and kind of prepare the immune system of a recipient to attack whatever you transplanted into the patient. Now, what are the factors that are contributing to this? We need to know the factors to know how to avoid them. So first of all is the methods of decellularization. The physical, chemical, and biological methods of decellularization all have these pros and cons, and the combination of them is that fine-tuning that we still have to learn how to do. Then the alterations of those proteins that we want to stay in a scaffold, because when we wash and process and keep it at certain temperatures and introduce some enzymes and stuff like that, we break those molecules in an unusual ways, creating some pieces that are also very, very upsetting for the immune system of the recipient. We also have to take in mind that some scaffolds are less immunogenic than others. Let's say when we're talking about the cyst, which is a pig gut, they tend to be much less immunogenic than, let's say, the heart valve or the liver. The last factor that contributes to it is where you're putting your transplant. So if you're putting it in a heart, there is a one reaction. If you're putting it on the skin, it's another. So as you can see, there is a whole lot that we still need to find out and address and develop in a very specific way. So the solutions, what can we do? In the recent years, there is one new approach to solve this problem. Is instead of taking already dead tissues that are going through the necrosis, initiate the programmed cell death, which is called the apoptosis. You probably never thought about it, but you all know that the embryos, when we develop, even as a human beings, we have these membranes between our fingers, right, while we're embryo. And by the time we're all born, none of these membranes exist. So those cells are not necrotizing. They're just sent into the programmed cell death and do not induce any inflammation. So that's why the scientists came up with this approach, because in this case, when your cell is dying by programming it, by organism telling it, you go die, but don't upset the equilibrium, what happens is that remnants of that cells are staying enclosed in membranes and not spilling all of its guts to expose the immune system, and then it all causes the inflammation. So that also produces much smaller pieces of DNA that are easily washed out, and all of this causes less inflammation and anti-inflammatory responses, but it's been done and was shown that it causes just quite as good decellularization as we do it with the washings. That's one. And another one, meanwhile, while we're perfecting and making the apoptotic design decellularization, we're dealing with what we have today by trying to remove selective antigens that are specifically annoying to the immune system of the recipient, by cross-linking some DNA pieces, by solubilizing insoluble proteins that we don't want to be in a scaffold, and eventually by treating our scaffolds by sterilization, so we would avoid opportunistic infections by viruses and bacteria that can induce an immune response in themselves. So with all this, I don't want you to get discouraged. In fact, the opposite. I want you to understand that the sky is the limit, and you have a lot to do before we'll have a ready solution for regenerating and replacing any part of your house that you need to become new and shiny. Thank you.